Welcome back to our bio series. Today's topic is one of the most exam heavy area, enzymes. We are going to solve first year bio chapter 3 MCQs with step by step reasoning and option elimination. Not only will you learn the correct answers but also the logic behind them. So stay till the end because I will also share a short trick that will help you crack any enzyme related MCQs and exam. So the question number 1 is the catalytic activity of enzyme is restricted to its small portion called. The options are the active site, allosteric site, Holoenzyme and substrate. So the right answer is the active site because active site is the small region of the enzyme where the substrate binds and catalyzes the reaction. Why the allosteric site is not the right option? Because allosteric site is the regulatory site, not the catalytic site. And holoenzyme is an entire functional enzyme, while substrate is the molecule acted upon, not the site. The next question is which of the following has coenzyme activity? So the options are the NAD, Ca, calcium, both and none. So the right option is NAD plus because NAD plus is an organic vitamin derived molecule which is a classic coenzyme. Why calcium is not the right option? Because calcium is inorganic and it acts as a cofactor, not as a coenzyme. So the right option is the NAD plus. The third question is non-competitive inhibitor react with enzymes 8. Active site, allosteric site, both A and B and none of them. So the right option is the allosteric site because non-competitive inhibitors don't bind at the active site, instead they distort enzyme shape by the allosteric site. So active site is for the competitive inhibitor. So the right option is the allosteric site. The next question is the combination of Apoenzyme and Coenzyme produces prosthetic group, Holoenzyme, enzyme and cofactor. So the right option is the Holoenzyme because the Holoenzyme is made up of Apoenzyme, which is the protein part, plus coenzyme or cofactor, and that Holoenzyme is a complete active enzyme. Now the question number 5 is the specificity of enzyme is due to their options are the surface configuration, pH, hydrogen bonding and high molecular weight. So the right option is the surface configuration because the specificity is due to amino acid sequence and shape of the enzyme and it determines which substrate will fit. Remember the lock and key model. Now pH affect the activity, not the specificity, and hydrogen bonding important but not the main reason of enzyme specificity. High molecular weight is irrelevant, so the right option is the surface configuration. The question number 6 is an essential feature of a competitive inhibitor is its ability to activate an operator gene, combine with prosthetic group, modify a substrate, or occupy an active site. So the right option is occupy an active site because the competitive inhibitors mimic substrate and block the active site. So the right option is the D option. The question number 7 is the reaction rate of salivary amylase with starch decreases as the concentration of chloride ion is reduced. Which of the following describe the role of chloride ions? So the options are the allosteric inhibitor, cofactor, coenzyme and competitive inhibitor. So the right option is the cofactors because chloride ions are inorganic helpers working as a cofactors. So by reducing its concentration, it will decrease the rate of the reaction. The question number 8 is how does an enzyme increase the rate of a reaction? The options are by bringing the reacting molecule into precise orientation. The second option is by increasing the rate of random collision of molecules. The third option is by shifting the point of equilibrium of the reaction. The D option is by supplying the energy required to start the reaction. So the right option is the A option. By bringing the reacting molecules into precise orientation. Why? Because enzymes deliver activation energy by proper orientation, not by collision, not by equilibrium shift, or not by providing energy. So the right option is the A option. The question number 9 is many enzymes are secreted in inactive form to protect. Options are the cell proteins, mitochondria, cell membrane, and cell DNA. So the right option is the cell protein because digestive enzyme could digest the cell itself. So they are secreted and inactive precursors known as zymogens. The question number 10 is Erypsin is an example of carbohydrates, proteases, lipases, and nucleases. So the right option is the proteases because Erypsin is a proteolytic enzyme which digests proteins. The question number 11 is ribozyme consists of options are only protein, protein plus non-protein part, only RNA, or none of them. So the right option is the C option, only RNA, because ribozymes are RNA molecule with catalytic activity, not the protein part. The question number 12 is if an enzyme solution is saturated with substrate, the most effective way to obtain and even fast yield a product is to add more of the enzyme, add more substrate, add an allosteric inhibitor, add a non-competitive inhibitor. So the right option is the add more of the enzyme. Its saturation enzymes are fully occupied. Only adding more enzyme will increase the rate. Now question number 13 is if an enzyme is added to a solution where its substrate and products are in equilibrium, what would occur? The options are nothing, the reaction would stay at equilibrium. The B option is additional products would be formed. The C option is additional substrate will be formed. The D option is the free energy of the system would change. So enzymes speed up reaction but do not shift equilibrium. So the right option is the nothing, the reaction would stay at equilibrium. Question number 14 is enzymes make it possible for cells to escape the need for energy? So no. Are non-protein molecules that help coenzymes? So no, because enzymes are protein molecules. The third option is are not affected by change of pH. So no, because pH can affect the enzyme activity. The D option is lower energy of activation. So yes, it could be the right option because enzymes are proteins that lower activation energy. So the D option is the right option. Question number 15 is an allosteric site on an enzyme is the same as the active site where ATP attaches and give up its energy, often involved in feedback inhibition. All of these are correct. So the right option is the often involved in feedback inhibition because allosteric site allow regulation, especially feedback inhibition. So the right option is the C option. The question number 16 is at high temperature options are can affect the shape of enzyme, lowers the energy of activation, makes cell less susceptible to disease. Both A and C are correct. So the right option is the high temperature can affect the shape of an enzyme. So the A option is the right option because high temperature denature enzymes. The question 17 is the coenzyme is often a metal, always a protein, often a vitamin, always an inorganic compound. So the right option is often a vitamin because coenzymes are organic, vitamin derived. Metals are cofactors, not coenzymes. So the right option is the C option. The last question is blocking of enzyme action by blocking its active site is called as feedback inhibition, allosteric inhibition, competitive inhibition, and non-competitive inhibition. So the right option is the competitive inhibition because the direct blockage of active site is the function of the competitive inhibitor. And that's it. We have just solved 18 MCQs on enzymes with explanation and elimination technique. Now here is one thing that I want to tell you, that in just video lectures I can't cover all the possible MCQs. So keep visiting YouTube channel community and take active participation in answering questions. Hopefully you will learn a lot. Now here is a quick exam hack for enzymes. Apoenzyme plus cofactor that make Holoenzyme. Cofactors are inorganic metal ions while coenzymes are the organic vitamin based. The active site is the catalytic site. A competitive inhibitor inhibits the enzyme activity by binding to the active site, while the non-competitive inhibitors inhibit the enzyme activity by binding to the allosteric site. Now, if you found this helpful, don't forget to like, subscribe and share with your friends. Until next time, keep practicing and keep learning.